Welcome, everyone. You're at Silver Hill Hospital Grand Rounds, and we'll get started here in just a moment as people settle into their Zoom boxes. All right. Welcome, everybody. My name is Jeff Katzman. I'm the Director of Education here at Silver Hill Hospital. I hope everyone has had a great summer. And today really marks the return of our Grand Rounds Fall series, where we are absolutely delighted to restart and welcome Dr. Matthew State, who will be presenting on the topic Autism Spectrum Disorder Genetics from the Research Lab to the Clinic. And I have the real pleasure of introducing him in just a moment. But first, some further information. You may have noticed a new logo on our invitations. We are excited and really proud to launch the Silver Hill Academy for Research and Education. So that spells S-H-A-R-E, so we call it SHARE. And all our educational efforts will fall into this newly formed SHARE umbrella, whose mission is to provide education and lifelong learning for mental health providers. It includes our continuing education programs, research, training programs for psychology and psychiatry and all disciplines, our community programming, our resilience work, and advocacy. So please refer to our website for more details about SHARE and how we're integrating our learning vision. Next, our next Grand Rounds will take place on September 27th, where we will welcome Silver Hill Hospital's own Dr. Elizabeth Ortiz-Schwartz, who will talk to us on social media use in adolescence. And you can sign up for this event on our hospital website under Education and Resources, and then the calendar. So now back to today's lecture. I am so delighted to be joined by Dr. Andrew Gerber, our President and Medical Director, who will co-moderate the Q&A session with me after Dr. State's talk. Your questions remain a really important part of our Grand Rounds. It's your opportunity to put forward questions and comments to Dr. State, and you can submit a question anytime during the lecture using the Q&A box. A key component of our Grand Round series is providing no-cost continuing education credits. So if you wish to receive CME, CEU, or contact hours, please complete the evaluation survey that will automatically show in your browser when we end the webinar. We will also email a copy of the survey to today's participants. And finally, disclosures. No planners of this activity have indicated a relevant financial relationship with an ACCME defined commercial interest. Dr. State will show a slide with his own disclosures. It is now really a great pleasure and honor to introduce our guest speaker today, Dr. Matthew State, who is the Oberndorf Family Distinguished Professor of Psychiatry and Behavioral Sciences and Chair of the Department of Psychiatry at the Department of Psychiatry and Behavioral Sciences, President of the Langley Porter Psychiatric Hospital and Clinics, and member of the Weil Institute for Neurosciences at the University of California, San Francisco. Dr. State is a child and adolescent psychiatrist and human geneticist. Over the past 20 years, his laboratory has played a leading role in illuminating the genetics of childhood onset neurodevelopmental disorders, including autism spectrum and Tourette disorders. He has been the recipient of numerous awards, including the Tarjan Award from the American Academy of Child and Adolescent Psychiatry, the Ruane Award from the Brain and Behavior Research Foundation, and the Rhoda and Bernard Sarnat International Prize in Mental Health from the U.S. National Academy of Medicine. He was elected to membership in the National Academy of Medicine in 2013. I will say also that I've known Matt for many, many years, all the way back to college. And we trained together in psychiatry and residency training back at UCLA. And I have always really known Dr. State to be among the brightest and most thoughtful and also kind of coolest people I've known forever. And I just want to say that UCSF is extremely lucky to have him at the helm. And really, we are quite fortunate that he said yes to the invitation that I sent out to speak to us today. So with that, I'm going to pass the baton, Dr. State, over to you, and Dr. Gerber and I will turn off our videos and we'll rejoin after your presentation for Q&A. And again, remember, you can type in a Q&A all along the way.
Great, thank you. Thank you, Jeff. I'm delighted to be here. I think it's the first time in maybe 3 decades that someone has used the word cool in my introduction. I'm glad that this is being recorded. I'm very happy to be able to join you. Today, I'm going to talk about autism spectrum disorder genetics, as the title says, from the research lab to the clinic, and I'm going to do my best to take a topic that I think is often opaque, the genetics of psychiatric and child psychiatric disorders, and hopefully make it less so. I'm glad that we have plenty of time at the end to have a Q&A, so that to the extent that I don't accomplish that goal, hopefully I'll be able to improve upon that in our exchange. What I want to do today is to set the frame. I am going to take a little bit of time to do some kind of genetics 101 for those of you who haven't looked at this since your time in training or as an undergraduate. Hopefully it'll be helpful to get us all on the same page. And for those of you who think about this all the time, you can make notes and help give me suggestions about how to do this better in the future. I'm then going to talk about sort of where the state of the science is now with regard to gene discovery in autism. Talk a little bit about what that tells us about the biology of autism spectrum disorders. And then in the last part of the talk, consider what this means for the clinic, both in terms of things like genetic testing, but also where this may be headed with regard to the development of new approaches to therapeutics. My disclosures: I'm a member of Scientific Advisory Board for MapLight Therapeutics. That's a startup biotech company interested in developing therapeutics in psychiatric disorders, but there's nothing about my talk today that directly overlaps, except at the kind of very highest conceptual level with the work that's being done at MapLight. I wanted to start with a little clinical vignette, just, as Jeff pointed out, as child psychiatrist first and geneticist second. Quite literally, I finished my clinical training and then went back to do a PhD in genetics in order to try to understand at a deeper level what I was seeing in clinic. But I think it's helpful to place this in a clinical context, and we'll come back to this towards the end of the talk to discuss why this was shaped in the way it was shaped. I do want to take a moment before we begin to talk about this and then start to talk about sort of genetics 101, just to make a comment about the fact that there's an important conversation going on right now around neurodiversity. And that includes, I think, a lot of important questions about how we define disorders within child psychiatry, obviously specifically how we define a disorder like autism, and where across what is an enormously broad spectrum of symptoms or clinical manifestations, sort of where we draw the line between human diversity and human pathology. This talk is explicitly focused on a subset of that experience that is defined in a kind of medical context, and so I'll be using terms like pathology and mutation, impairment, symptoms, and I want to say that just for we have a big audience here today, and just want to make it clear that this is not a way of painting all of the experience of the broad range of individuals who either carry a diagnosis of autism or have autism symptoms, to paint that entire population with a broad brush. In fact, this is, as you'll see through the course of the talk, a lot of the progress in genetics is has been focused on the intersection of autism with other neurodevelopmental disorders, including epilepsy and intellectual disability, motor development, et cetera. And I think while it is could take up an entire lecture to talk about how we think about distinguishing between the areas that we, define as pathology and those that are not. But we won't delve into that in great detail today. But I did want to give that preface. As I said, I think it's a very important conversation, and while the talk will focus on autism as a disorder, again, that is not meant to categorize everyone who experiences variation in reciprocal social communication or their range of interests with that broad brush. Okay, so with that said, we're going to focus on ASD, and you now I think probably have had plenty of time to read this. This is an 18-year-old young woman who's being treated for anxiety and depression, carries a diagnosis of ASD, does not have intellectual disability, does not have epilepsy, has no family history of ASD, and has never had genetic testing. And so a question that might come up is whether or what type of clinical genetic testing should or could be performed on this patient. And as you read through this, for some of this audience, this may these acronyms may be impenetrable, but please, my hope is that they won't be by the end of this talk. There are obviously multiple choices here, whether or not any is indicated, because this individual does not have the kinds of comorbidities that I just mentioned around intellectual disability or epilepsy, any evidence of brain malformation. Their question, B raises the issue about whether or not we should identify SNPs and determine polygenic risk. And again, before the end of this talk, that should be transparent in both what those terms mean and what the answer should be today, given the state of the science. And then questions about whether or not you should look for, use a chromosomal microarray to look for submicroscopic changes in chromosomal structure, or do more extensive sequencing. And then this last question is whether or not any of this or this can really be understood without understanding the ancestry of the patient, which again, I think is another really important conversation, not just in autism, but in all of human genetics. So we'll come back to this at the end. And now what we're going to do is launch into genetics and genetic variation 101. Again, this is meant really to try to get us just on the same page with regard to the terms and concepts that are essential, I think, to understand what has happened and what is happening in the field. So the first thing that I want to remind you about is just DNA, and that the human genome is composed of DNA. Every cell in the body that has a nucleus has all of the instructions in the DNA that are required to drive biological processes and lead to the human development. There is what we call the central dogma of molecular biology. So the way that DNA, which can be thought of as sort of a set of plans or a template, the way that that's translated into biological processes is through, again, what we call the central dogma, which is the DNA is transformed into RNA, and then RNA, in most instances or many instances, is turned into proteins, which are sort of biological machines that drive forward biological processes. I just gave a bit of a caveat. Not all RNA is turned into proteins. There are RNAs that directly impact biological processes, but that sort of overall framework of DNA to RNA to protein is something that really undergirds molecular biology and a lot of what we're going to talk about today. And then the term that we use to talk about that process of going, particularly from DNA to RNA, is transcription. Again, I'm sure all of this is familiar to many of you, but it just, if you haven't been thinking about it during your clinical activities over the last months or years, just a brief refresher here. So that's the substrate that we're talking about, which is the human genome and DNA. What we're going to talk about is the extent to which we're able to find evidence of risk within the DNA, the human genome, that contributes to or leads to autism. Before we do that, I want to talk about the concept of genetic variation. So when I'm in person, at this point in the talk, I would typically say, look to your left, to look to your right, regardless of who's sitting next to you. At the level of your genome, you're about 98.5 to 99% identical to that person. And then in the day, I would make a joke about always wanting to sit next to Eric Kandel. On video, I guess we're on, this is a webinar, so you won't be able to look in the Brady Bunch squares, but I think it's an important place to start. As humans, we are far, far more like all humans than we are different. And again, at the level of DNA and our genomic DNA, we're between 98 and 99% identical to any other human. The reason that I want to focus on that is because the extent that we are interested in how genetics leads to pathology, we're not interested in that 98 or 99%. We're interested in how the genome varies between individuals, and particularly how the genome varies between individuals who have a given disorder or not. So we're going to focus for the rest of the time here on genetic variation. The rest, that, the issue about how the genome works, its biology, ancestral history, all incredibly interesting, and many Nobel Prizes that have emerged from the study of the 98 to 99%. But we're going to be focused on the 1 to 2% that varies. And over the last 15 to 20 years, and particularly with the sequencing of the human genome at the turn of the millennium, we've learned a ton about genetic variation, which is probably the most important point that I will make in terms of what has allowed us to move from an era of essentially knowing almost nothing about the genetics of autism to now knowing, as you'll find out, quite a bit, with several hundred genes that we can confidently say are associated with varying degrees of risk for autism spectrum disorders. There are two types of genetic variation that I'm going to talk about in more detail today. And again, hopefully this will be a helpful refresher. So DNA between individuals, that 1 to 2%, that variation can happen in a variety of different ways. One way that we're often used to thinking about that is just that the letter code of the DNA, I didn't mention, but I think most of you will recall from your basic biology courses that DNA is made up of 4 nucleotides that are abbreviated A, C, G, and T. And between any two individuals throughout the genome, you will find, again, in this kind of 1 to 2% of the genome that varies, at any given spot that you look in the genome, that there can be changes in that letter code. That is a normal part of human variation, helps drive forward human evolution. And I'm going to come back to talk in a little bit more detail about how to think about sort of differences within that category of what we call sequence variation. So one type of variation we're going to talk about is sequence. You're seeing a picture there. The other that we're going to talk about is variation in the structure of chromosomes. Chromosomes individually are just one long piece of DNA, actually wound around other proteins. Often thought of, like, as this picture shows, as kind of these static bar-like creatures. In fact, it's highly dynamic and an enormously interesting area of study to understand how the 3D conformation of DNA has an impact on biology biology and how epigenetics impacts that process. But what I want to talk about in depth today is the notion that in addition to the DNA being potentially varying at any given letter in the DNA code, we've also found that that the structure of chromosomes vary between individuals. So this is an old enough picture here. I think it's quite useful to see the ways in which that structure can vary between individuals. So you can lose genetic material, you can gain genetic material, you can take a piece of genetic material and flip it. But one of the important discoveries shortly after the human genome was sequenced was that these kinds of structural changes actually are quite frequent in the genome and are not necessarily abnormalities. So at a time when we could only visualize the genome under the light microscope, if we saw one of these things, a deletion, a duplication, or inversion, that was sort of implicitly thought of as being related to pathology. When we were able to look at the DNA at much higher resolution, we found it's replete with sub-microscopic variations in chromosomal structure, again, losses, gains, and changes in orientation. That group of variation turned out to be an entree into understanding the genetics of autism spectrum disorders. So I've told you about sequence variation, told you about structural variation. I'm going to come back and talk to you about findings in both of those areas and their contribution to autism spectrum disorders, and I would say to a whole range of neurodevelopmental and psychiatric disorders. In autism in particular, there's a type of change in both sequence and structure that has turned out to be quite important for understanding the genetics of autism and autism risk, and also turned out to be a particularly important avenue to discover specific genes related to the syndrome. So this is variation. It's called de novo variation. Typically in many, perhaps most, discussions about the genetics of psychiatric disorders, people would sort of automatically think about the transmission of genetic information from one generation to another around heritability. It is the case, and we're going to come back to this, that for many psychiatric disorders, risk is carried from generation to generation, including in autism. We'll talk a bit more about that. It's also the case that as a normal process, in every generation, a small amount of new mutation is generally introduced to any genome. Now, that can be introduced and have no effect. It at times can be introduced into a genome and have considerable effect. It was a focus of my laboratory from early on to try to see whether or not we could leverage that process, new mutation and in the genome, which, as I said, it happens at a low rate in each generation, so new mutation is quite rare. So the focus of my laboratory from its inception was, could we use rare and new, or called de novo variation, to try to understand some portion of what was going on in autism spectrum disorders. And so the reason that that was at least conceptually attractive is that, as I said, de novo mutation is introduced newly into the genome in every generation. If that happens in sperm or egg just prior to conception, what happens is that it's then present in every cell that has genomic DNA in the offspring, and it is not present in the parents. And if you think about it, that offers a potential window into genetic mutations that could have potentially large effects, meaning that some genetic variations that could cause serious disorders that over time would be weeded out of the genome, that the de novo mutation in the generation that it arose potentially could be observable if you had the technology to do it. And, you know, I've always said it's better to be lucky than to be good, hopefully best to be both. But one of the things that was very lucky for our laboratory is that we had an interest in de novo mutation. When I started my lab, the only way that we could find this was by looking under a light microscope, and then the genome was sequenced, and a huge array of technology developed that allowed us to focus on this new mutation as a potential contributor to autism. So in fact, it's turned out, I'm going to jump now over, almost a decade of toil and failure, and take you to success, which is that we actually determined that de novo variation, both in the sequence and structure of DNA, plays an important role in autism. I'll help define that further, but to say that those mutations can, in certain circumstances, carry very high risks for autism, and for other neurodevelopmental disorders. And just want to give you a little bit of a sense about sort of how the study of de novo variation allowed us to make discoveries around specific genes. So the first thing is that I said the genome is a very big place. I didn't mention it, but there are variations in the genome at the level of sequence in particular that are extremely common. In fact, anything that is present more than 1 to 5% of the population is called common genetic variation. And there are millions and millions of those variations. There are very few in the portion of the rare de novo mutations in any generation. So in the portion of the genome that makes genes, that's only about 1% of the overall genome. There's essentially about one of these events per generation. And so I have a picture here of a forest with a contrasting tree, just to point out that in looking for things that might be related to autism, our approach in the laboratory was highly reductionist. It allowed us to really only need to count to one, essentially. So we could look through the DNA of individuals with autism and see whether or not there were children who were carrying a new mutation that was not present in their parents. And then we could ask, as this diagram shows on the left, whether or not mutations in that gene were more often found in a child who had autism than in their unaffected siblings. Now, that provides not only, I think, a relatively easy experiment compared to taking a look at huge populations and trying to see, how many thousands of changes might need to conspire to increase the risk for autism. It applies to only a subset of individuals with autism, but it allows for this high level of contrast. And then I also want to point out that that it provides a tremendous amount of statistical power. And the reason is again, that the genome is a very big place. It's got 3 billion bits of sequence information. The part of the genome that codes for genes has about 20,000 genes, constituting about 3 million bits of information. And in any one generation, in a random way, a new mutation may be introduced. Across the entire genome, we know that between 50 and 100 are introduced in the portion of the genome that we understand best, the part that makes genes. Again, it's about at most 1 per generation. So when you think about that, you have this enormously broad search space. You have essentially a genetic lightning bolt in a family. And then the issue of whether or not, if you look through a whole bunch of other people, whether you would ever see that lightning hitting the same tree again, actually gives you enormous statistical power. And you very quickly can identify among groups of individuals that if you see what we call a recurrence of a de novo mutation, and particularly a de novo mutation that we can predict is going to have an impact in a negative way on the translation to RNA or the production of a protein, that gives you enormous statistical power then to be able then to say that is not just a single random lightning strike, but it's accumulating in individuals with autism, not accumulating in their siblings without autism. And that gives us an ability with a lot of confidence to say that, that these genes are related to ASD. And so it turns out that autism is that was a valuable, important way to look for mutations in autism spectrum disorders. And it turns out that among particularly adult onset psychiatric disorders, this is a pretty distinctive genetic signature. So autism is distinctive among neuropsychiatric disorders to date for the identification now of more than 200 genes that, when they are subject to these rare de novo mutations, and particularly those that damage or eliminate the protein that would be encoded through that central dogma that I talked about, that those are contributing and as we anticipated, they might carry very large biological effects compared to what you would typically hear about in a genetics of psychiatry lecture, which are at the other end of the spectrum, sequence variations that are common in the genome. And what we know about those is that overall, they were expected and have turned out to individually carry very small effects. And that's what this figure is showing you. So this is a snapshot of gene discovery to date for autism spectrum disorders, contrasting with gene discovery to date in schizophrenia. What you're seeing on the vertical axis is the relative risk or the effect size. This is in fold effect. And then along the horizontal axis, you're seeing how common are these mutations found in the human genome. And what you can see in autism spectrum disorders, as I said, is that these rare mutations, and particularly de novo mutations that damage the protein, we call them de novo damaging variants. And by the way, that can be sequence or structure, that they carry very large effects. And that's been where the lion's share of gene discovery has been in autism. And then you have this inset that begins to show you that this is not just a kind of a general description, but because of that issue of rarity and recurrence, we can tell you what the specific genes are that are being hit by these de novo mutations and that they are carrying risk. Now, conversely, in autism, you can look to the right, and those blue dots are what we found so far with regard to common variation in the genome that's present in more than 1 to 5%. And those kinds of variations in the genome carry exceedingly small individual effects. This is true across all of psychiatry so far, as we've been able to tell. There are few examples where common variation carry very large risk for psychiatric or So now, conversely, in schizophrenia, what you can see is that those small effect common variations have been the lion's share of discovery. So now the latest papers are hundreds of these individual variations, each one contributing very, very small effects. Very recently in schizophrenia, after sequencing thousands and thousands of individuals, we began to see that there are, in addition, these rare mutations that also increase risk. But this does give you a sense as to paradigmatic disorders, kind of how autism right now differs. And so when people, talk in an offhanded way around about genetics of psychiatric disorders, often you'll hear someone say, well, it's highly polygenic, and it's a conspiracy of common genetic variation, and the way that we need to look at this is by summing all of that in a polygenic risk score. And all of that is a good general description of the genetics of psychiatric disorders, but it is also the case that ASD is distinctive, again, in having these very large effect clues at the molecular level about the genetic risks for ASD. I'm not going to belabor this because I realize I've taken longer on giving you the background and genetics of autism than I initially intended. I hope that it has not been repetitive for many of you. The only thing that I want to point out is that that picture of gene discovery is accurate, but it is not a statement about the population risk for autism. So if you read an autism genetics review, the first thing that you will read in many would be that autism, like schizophrenia, depression, anxiety, that population risk is carried in these small effect common variations. And that is true. And the issue is that most people who carry that risk will have no symptoms of a psychiatric disorder, or in this case, no symptoms of autism. So that means that within a population, there can be a ton of risk that never makes it into your clinic. And what this diagram is meant to show is that these de novo, the LGD is an abbreviation for likely gene disrupting, another way of saying a damaging mutation, one that is expected to interrupt the normal process of a gene becoming RNA, becoming a protein, that while those are rare, and now in our genetic testing, we find that in broad populations, about 10% of individuals who come into the clinic and get tested for autism spectrum disorders will have a finding in around a rare mutation, often de novo mutation. That number varies depending upon the clinical population. But again, the notion here is that while in the population they may be rare, they carry a lot of risk. And what that means is that they're highly enriched in your clinical population compared to in the general population. So the only point that I wanted to make again is that the ideas that may seem contradictory, that most of the genetic risk for autism is carried and transmitted from generation to generation in this polygenic conspiracy of risk, that in fact in the clinic it can be a much higher representation. I said 10% for the very broad population. Within subpopulations there, depending upon how you define the clinical population, that number can go north of 50%. So if you're a medical geneticist and you're seeing someone who's been evaluated, has a diagnosis of ASD, with or without intellectual disability, if there's any concern that there may be features of dysmorphology or other comorbidities like epilepsy, that will enrich for finding these de novo variations. As a general proposition, as I said, in an all-comers kind of psychiatric clinic, if someone is diagnosed with ASD, you've got about a 10% chance, if you do genetic testing, that you will find either a change in the submicroscopic structure of the chromosome, that's called a copy number variation, or one of these known autism risk genes. So that means 90% you won't. And depending upon how you stratify the population, I should say, in addition, girls are much more likely, if they're diagnosed with autism, to carry one of these de novo mutations than boys, again, if they're in the clinical population. Okay, so I'm going to move on now to talk about what this means for us. That I hope I've convinced you that, despite sort of, I think, some nihilism at the turn of the millennium, that we would ever find genes of large effect for complex, common psychiatric disorders, that in autism now actually we're replete with these molecular clues. And so what do they tell us about biology? The first thing that we can say is, from the very beginning, as soon as we identified the first genes by using the strategy of looking for these de novo mutations of large effect, honestly, even in the first 3 genes, one of them was involved, at least overtly or prima facie involved, in synaptic function. One was involved in the regulation of that process of gene expression, meaning that central dogma, the genes going from DNA to RNA to protein. So that process of gene expression, the regulation of that, particularly through the modification of these proteins that make up something called chromatin. So out of 3 genes, one was synaptic function, one was chromatin or gene regulation, and a third was not. And an important reminder that nature doesn't carve at the joints. But what's happened since then is that, as we went from 3 genes to 10 genes to 71 genes to 102 to now 255, is that sort of general gestalt has held up. And a large proportion of the genes that we discover have already been studied and have been and are known to have function at the synapse, and another subgroup have been studied and are known to be involved in epigenetic regulation and the regulation of gene expression or transcription. Now, I have to say that starting in this field when we knew nothing about this, being able to show you this diagram still is gratifying. But I'm also mindful that essentially what we've done is an enormous investment in the genetics of autism. And we've gone from telling you that autism is a neurodevelopmental disorder, identifying genes, and now we're telling you that the genes are involved in the neuron and in development. So I think it's a fair concern that this is an extraordinarily complex but penetrating glimpse into the obvious. This is a neurodevelopmental disorder. I think that's true, and it's an important critique that I think has driven a lot of our work in the lab to go beyond that kind of generic statement about what's going on in autism, so that we can get hopefully to a level where we can say enough about it to begin to identify whether or not there are opportunities to develop treatments as a consequence of this understanding of biology. So you would think that the way that we would do that, and this is sort of the standard model, translational model for years, before we found genes, I would put this up and say, this is, kind of what our aspiration is. So we find this single rare mutation, and I should be clear, I talked about rare mutation and about the fact that they have very large effects. All of the evidence to date suggests that in any individual, that kind of event, a single event in the genome is carrying that amount of risk. So that may interact with other changes in the genome, but it really is the case that we're identifying specific changes at the molecular level that are, if you remember that distribution from sort of no symptoms to the clinic, a single event in the human genome is in many individuals is pushing people along that continuum. So you think, okay, well, if you know that, then if we can trace how the DNA goes to RNA, how the RNA goes to a protein, then potentially we could understand what cells, or we have a picture of a neuron here, could be a neuron, could be glia, but what cells in the brain those proteins act in. And then we could potentially understand what circuits those were involved in, and then we could, pull on that thread and understand something about behavior. And really, this notion that we could go from genes to behavior is encapsulated in this highly simplified notion. What I would say is that prior to gene discovery, we actually thought that might work. Then we discovered the genes, and when you discover the genes, you're immediately reminded of all the other stuff that you learned about biology that while we were so busy trying to identify the gene, we sort of conveniently forgot. So which is that while it may be a single gene and a single event that's increasing risk, genes along that continuum from genes to RNA to protein, it's not a one-to-one relationship. So one gene can make a variety of different RNAs that are related to each other but not identical, and those can lead to a variety of proteins. And so that is what is shown in moving from the kind of first level of this diagram to the second. And then we know that particularly in the brain, that is a highly diverse cellular architecture, that those proteins may be expressed in a whole variety of different cells, and that those cells are going to contribute to multiple different circuits, and those circuits, as we found over time, don't carve at the joints either, so that they subserve often over time a broad range of behavior. So this idea that there might be this direct line, even from these rare mutations of large effect to biology, is really a one-to-many problem. And then as we in autism, this is happening over development and not in cross-section. So maybe in cancer, that would be the level of complexity if you have a de novo mutation, not in the germline, but in a cancer cell. And but in our world, this is taking place not only in what we call this kind of biologically pleiotropic environment, but that's evolving over time. And it's particularly dynamic during early development. And so what we've got is like a single gene leading to multiple different proteins in multiple different cells, and that those things are changing over time. So you have, even in what we had hoped might be a single thread that you could pull on, a one-to-many problem, and almost an infinite search space there. And then we have another kind of challenge, which is that, the kind of standard way of dealing with genes of large effect would be to put them in a mouse and then see what happens. And that certainly that can work, and there's important progress going on in that area. But as a general proposition, those kinds of approaches that for disorders that have distinctly human manifestations, like alterations in reciprocal social communication and social salience and language development, et cetera, that we run into this problem that sort of the further we get away from the gene, the less we can rely on the mouse brain giving us kind of reliable insights into what's happening in human brain. So one of the ways that we decided to approach this, I think, may seem, obvious. It was in retrospect, it wasn't prospectively, but the notion was, and this has been picked up, actually it started implicitly, if you hark back to my discussion about are thinking about what do these genes do, what we were essentially doing is saying, well, we've got more than one gene, so maybe we can begin to categorize. And that sort of process actually it lays out sort of a strategy to say, well, if you have 71 genes or 250 genes that are all contributing to autism, somewhere along this continuum from these genes of large effect to human behavior, they're likely to converge. And so the one way to begin to think about this would be to ask the question, again, as we did implicitly at the start, sort of, what is similar, and are there subgroups that that we can find that are reliably statistically different from what you would expect in the null hypothesis under normal development. And one of the things that we could do is that we could do this not only with regard to what do genes do and say, well, is there a way in which their function is similar, but we could also begin to ask the question as maps of the molecular development of brain emerged kind of simultaneously with advances in genetics, that we could begin to ask, is there a point of convergence around when and where this is happening, and not just kind of what's going on, as I said, kind of, with these ideas that it's the synapse or regulation of gene expression. And we're really surprised to find out starting 10 years ago when we first did these experiments, and now this has been borne out multiple times across many laboratories, is that the answer is, yeah, you can, if you take the top genes, and whether we started with 10, as I said, we've moved our way now all the way up to, more than 200. And you do begin to see that not only are these coalescing around certain functions, but they're coalescing around particular points in time in which this risk is emerging. And the bottom line for this is that there's a tremendous coalescence in fetal development, and particularly in mid-fetal development. What we know most about right now, and in part this may be a result of the underlying data, which is richest around certain brain structures like the cortex, but there's clearly very strong replication that the risk for ASD from large effect de novo mutations coalesces in early to mid-fetal development. Not only that, but we could begin to say where in the cortex is happening. So the cortex is a layered structure, and we could begin to tell you what cell types and what layers. And so ultimately, actually, I'm going to skip over this because of in an interest in time. Again, just wanted to point you to the fact that we don't know how long this risk window continues, how plastic it is. But we do have very strong evidence now that that the set of autism risk that is carried in these de novo large effect mutations is emerging in mid-fetal development, impacts multiple brain regions, but with a strong convergence in cortex. Now we're beginning to also see convergence in striatum. And as I said, we can tell you that there's a particular type of cell, an excitatory glutamatergic neuron. And moreover, we're beginning to gain traction on understanding where in the stages of development of those cells that autism risk is beginning to emerge. And that is sort of in neuronal proliferation of these of excitatory and in some cases inhibitory neurons. So that was a long-winded tour through what we know about the genetics of autism now. I do have to say that I'm going to take the last bit of the talk and then tell you about sort of how this is playing out or potentially plays out in the clinic. I'm just going to pause and say that for a scientist, what I've just shown you at some level feels breathtaking, that we've gone from literally understanding nothing about the specific pathology, again, of a subset of individuals with autism. This is not a broad brush about everyone with social disability, but particularly with people who carry these large effect mutations, that we can tell you a cell type, that we can begin to tell you what the proteins are and beginning to see how they interact with each other. That said, I'm also a clinician. As I said, I'm a clinician first, and I think that that sense of of excitement about elaborating autism risk in a way that we'd never been able to see it before. That there's also a sense of frustration and disappointment with what's happened with that knowledge over the decade now that we've understood how to find genes for autism and have been accruing these very strong molecular signals, because the take home of the last portion of this talk is that there has not been to date significant impact in the clinic. We have not impacted the lives of those who experience autism as a disorder or their families, and that is really an important focus moving forward. I worry that sometimes that, science takes a long time, and, we can see in other areas, we see cancer, immunology, that over time, the kinds of things that I've described for you already do transform or have the potential to transform clinical care in profound ways. If you think about pediatric cancers, the transformation of survival in large part has been predicated on the kind of molecular understanding that we're beginning to have around this subset of individuals with autism spectrum disorders. Definitely not guaranteed that we'd be able to do the same thing with a complex developmental brain disorder. But I think it does suggest that nihilism, because we haven't made more progress in the clinic, I don't, at least certainly in my view, I'm an inherent optimist, but couldn't keep doing this job if without really having a strong inclination, which I'm going to show you now, I think is supported by where things are moving in the clinic, that this can have an impact. I'm just going to say briefly around clinical testing, because I do want to have, I may run over slightly. I was trying to do this in exactly an hour, but I want to talk about that idea about whether or not molecular clues can lead to novel treatments. But clearly right now, the way that genetics for all psychiatric disorder is beginning to come into clinic is through genetic testing. So just this slide will, will be available to you to go back and review. But all it is really in many ways is summarizing that, yes, both common and rare changes in the genome can contribute to autism. What we know about specific genes contributing to autism resides in those rare mutations, so that genetic testing for common variations in autism right now has no clinical utility. And you may already know, I should have mentioned it at the time, but this abbreviation SNPs is single nucleotide polymorphisms. That term captures the notion of common sequence variation. So these individual common sequence changes carry vanishingly small risks. Even when you aggregate them, they can be useful to think about research, but are not useful in the clinic for any kind of prediction. Now, these rare mutations carry very large effects. What I didn't spend a lot of time on, but has been, in addition to all the complexity I've already described, has added an additional level of complexity, is while these genes clearly increase the risk for autism, it also turns out that if we take a cohort of individuals who potentially, say, have epilepsy without autism or intellectual disability without autism, we see many of the same genes. It's not completely identical, but there's enough overlap that it is very difficult right now to take a look at any one of these individual genes and say, we know that if we see that mutation, that the only thing that it is leading to or will lead to across development is autism. So that obviously poses considerable challenges for using early genetic testing as a diagnostic. If you have someone with autism, you can go back and you see the gene, you say, yes, that's one that is known to carry risk. But this obviously raises questions about, well, it actually doesn't raise questions, just want to say not useful for diagnosis. Okay. Now, that doesn't mean that testing for rare genetic variation is not useful. It does not carry a lot of clinical information right now. There are very rare circumstances where it does. I've mentioned one here, which is that one of the rare variations we've known about for an awfully long time, but turns out at times to show up in individuals who have not been identified for the classic features of Fragile X. But there are a handful where knowing that you have the mutation provides important information, either about reproductive planning, family planning, or potentially about clinical course. What has happened already is that as these genes have been discovered, families often want to know whether or not there's a genetic mutation in the family. And it's, I think, an important and worthwhile discussion with families who would like to know. One of the benefits has been that many groups have arisen in the advocacy community around these mutations, and that offer a variety of support, information, and connection with research, et cetera, to individuals who carry mutation in a specific gene. There are actually, large research studies that not only identify genetic mutations, but then help bring people together who carry a mutation in a specific gene. And for many families, that is considered to be valuable and worth going doing testing. In fact, the American College of Medical Genetics recommends that you look for submicroscopic changes in chromosomal structure for anyone with ASD who's not had genetic testing. Other professional organizations have not gone that far, say that should be done in the context of intellectual disability or other comorbidities, et cetera. I think it is a harbinger for the fact that this is going to become more and more common in clinical testing. Okay, so where I want to spend the last since I had an introduction, I'm going to go to 9:35 and then hand this back over. Is to talk to you about actually what's already happening in this world of large effect rare mutations in neurodevelopmental disorders that is now imminently arriving for individuals who may also have autism symptoms. So what you can see here in the diagram is this picture, again, showing this tremendous complexity that the minute that you move away from the gene, you're into this incredibly complex search space, so that, even if you mutate a gene and you see that it impacts a cell, knowing whether or not that is the thing that makes it all the way through this gauntlet to impact the behavior that symptomatology that you're interested in is a very challenging task. And so, not surprisingly, given advances in current technology, what this has done has been to place an emphasis on seeing whether or not you can go close to the gene, meaning that if you could correct at the level of the gene, even before the transcription or the translation or in that process, that would be a potential way to treat without having to move you into this highly complex biological space. There are multiple ways that we can do that now. Again, I'm not going to run through this entire slide. I just want to point out that there are ways either to directly target DNA or RNA in a way that alters its expression transiently, or there are ways to insert a normal copy of a gene if there's an abnormal copy of the gene that don't require you to go in and edit the germline genome of an individual. And that, I think, there's no question that the ethics of going in and directly gene editing in vivo is a huge ethical question and challenge. And so what I'm going to talk about now is some progress that has already been made and is being made around these genes that increase the risk for autism, but also increase the risk for epilepsy in certain circumstances, also increase the risk for intellectual disability, that the technologies that are being used to target these fall into these categories that are not editing of the patient's inherent germline genome. Part of the reason that this has been so of such interest is because of an absolutely transformative landmark finding that was started a decade before this, but was really signified by multiple papers in the New England Journal in 2017, showing that with a gene of large effect that was leading to a very early onset neurodegenerative disorder, spinal muscular atrophy, that you could use the strategy either of replacing the gene or of altering transiently gene expression through something called ASOs, which are synthetic pieces of DNA And I won't go into that and into how they work further. I'm happy to in the Q&A if people are interested. But the bottom line is, again, both of these technologies don't edit the genome themselves, but have been shown in spinal muscular atrophy to be able to be delivered in these cases through the CSF, to have penetration into the brain and to profoundly alter the course of this disorder. There are these leaves a variety of other questions. Spinal muscular atrophy is a central nervous system disorder, but it affects motor neurons. The question about whether or not other types of neurons and this can be done, et cetera, still remains an open question. But the underlying idea that for a very early disorder where a gene is identified as the symptoms are unfolding in the first year of life, that there are safe and viable strategies to do gene or nucleic acid targeting that, as I said, have transformed the clinical prospects for many children with spinal muscular atrophy. Conceptually, the way that approach, that those approaches are already being applied to what we call monogenic neurodevelopmental disorders like Angelman syndrome. Again, these nature does not carve at the joints, and we know that Angelman, when affects a gene that also is found at times with mutations in individuals who have autism without evidence of Angelman syndrome. But for a variety of complicated reasons. But the bottom line is that there are a number of what have been kind of longstanding recognized genetic syndromes where this strategy is being applied, and that set of genes not only to some degree overlaps with autism phenotypes, but the set of genes that we've discovered, and I'm going to skip over this in the interest of time. This is a table that is going to help me make my point that there are these what we call syndromic genes where these strategies are already in various stages of development. In Angelman, there are already clinical trials using these ASOs, or these short bits of synthetic DNA, to target the RNA in cells and see whether or not, if you do that very early in development, that you can alter the phenotype. But it's also the case that this is a set of genes that have been discovered in autism, and this list is growing, where the same strategies are being developed and will, I think, almost undoubtedly will begin to see them in clinical trials. One thing that binds these genes together is that they sit at this interface between features of autism and things like intellectual disability and epilepsy. And it undoubtedly will be the case that the therapeutic efforts will be targeted almost undoubtedly not as a primary endpoint at social development, which is extremely challenging to measure at any stage in human development, but including in the first months of life. But it is the case that as a consequence of these also leading to significant intellectual disability in certain circumstances, and to in some cases treatment refractory epilepsy, what you will see is clinical trials targeting the genes that we can also call autism genes. And what that will, I think, undoubtedly become is provide an opportunity to see what happens, and if these effectively target those comorbidities, what does that tell us about the ability also to alter in a positive way social development or other features that can accompany severe and profound forms of autism. Now, there are many challenges here, and we may get into this in the Q&A. I've already told you there are highly variable outcomes to these mutations. So again, the notion that you would find a mutation, not yet have a clear diagnosis of autism, and go in and treat with a genetic therapy is not on the table. It shouldn't be on the table at this point. There's not a one-to-one relationship. There are some cases in which people will carry these mutations and have either very mild or no discernible phenotype. So that poses challenges in thinking about how you would, beyond these kind of syndromic forms of intellectual disability and autism and epilepsy, how you would move forward. We clearly, I spent a lot of time talking about understanding where these molecular changes are beginning to have an impact or are converging in human development. That doesn't tell us how long we have. If the window opens and closes in midfetal development, obviously that's a huge challenge, both from a practical and ethical standpoint, but it's also not a foregone conclusion. I mean, we know that these things began to emerge in midfetal development in this group of kids, but there's nothing to suggest that, like other children with ASD, that there's not tremendous plasticity during early development, and that there are behavioral approaches and OT, PT, speech and language, et cetera, that can alter developmental trajectory. So the idea that there's plasticity there, I think we're seeing that, and we don't know anything about what we're targeting. So the notion that we actually would know specifically what we're trying to target, actually, I think is at least a cause for some optimism around the notion that even though we see these emerging in midfetal development, that the window is not going to be slammed shut then. There are a variety of regulatory issues. And then the last thing that I will say, and then I'm done here. Actually, I'm not, because I realize I have to take you back to the clinical vignette, but we can do that quickly. Is that a huge obstacle, which was kind of woven in this last part of my talk, is that we have a terrible time measuring the core features of autism, apart from being able to use that as a categorical diagnosis. For those of you who know about, the FDA approval of medications for autism, they're based on the aberrant behavioral checklist, which could not be sort of a cruder way of trying to capture irritability, et cetera. We don't have measures of social development now in early development yet that allow us to track that with the kind of precision that and dynamism that we would really need. We don't have reliable biomarkers, apart from being able to say that, if this gene has altered expression, we can fix that. But anything beyond that in a cascade, we don't. So a huge challenge for the field, now that we've had this tremendous advance, I think, at the molecular level, is that it really requires a Manhattan Project in the clinic for us to understand normal development differences in trajectory and underlying clinical, it's not underlying, and the clinical phenomena, particularly of autism on the severe and the pathological end of that spectrum. All right, so I'm just going to come back and say that I'm not going to belabor the clinical vignette, but just hopefully at this point, you would know that the answer to that depends a bit. According to the American College of Medical Genetics, you would definitely want to do genetic testing. I didn't mention, but I will now, that even in girls, for anyone who has not been tested, it is a strong recommendation to do Fragile X testing regardless. So there is some genetic testing that would be indicated, and perhaps for idiopathic ASD, looking for these rare mutations. But the part of what this is pointing out is that the absence of severe intellectual disability, epilepsy, or other features does not tell you what the yield of genetic testing is going to be in any specific individual. There's no cause for a genotyping array. SNPs or polygenic risk has no role in the clinic. Chromosomal microarray is recommended by the American College of Medical Genetics. Increasingly, I think this will continue to be recommended, but as we discussed, talking with a family about what the potential value of doing that is going to be important. And then at this point, whole exome sequencing, which is basically just a way of capturing more information about rare genetic variants, is coming along. It has not yet made it into clinical practice, but almost undoubtedly will as a substitute for these older generation tests. And then the last point is that it is true, and an important observation, that for common genetic variation, most of what we know is from studying Caucasian European populations. what that means is that in the research context, looking for these common variants and thinking about polygenic risk, that we have to be really mindful that that may only be relevant for for a small set of or a small subset of the broad worldwide population of individuals with autism. But again, those kinds of tests don't have clinical relevance at this point. That is going to be it so that we can move on. It takes a village. Often people just give a list of names. I always look up and, unless I can recognize one, it doesn't mean a lot to me. So I thought, maybe just showing the pictures of the wide variety of folks that have and continue to contribute to the work in the laboratory is here. And I'm going to end there and pass this back to Dr. Katzman.
Dr. State, thank you so much for a tremendous presentation, and wow, a whirlwind, and what you've done and what your lab has done is just remarkable. And thank you for presenting the information and really a glimpse into kind of your work and understanding of the developing understanding of genetics and ASD. I'm going to turn to Dr. Gerber for some remarks and maybe an opening question, and then I'll turn to the chat. And so please we have 14 in there already. So please feel free, everyone, to add questions into the chat.
Great. Well, thank you, Jeff, and thank you, Matt, for just a tour de force explanation of what is, of course, a complicated subject. So I'm going to be very brief because there's some wonderful questions out there, and I couldn't possibly do justice. But I wanted to start off with a maybe a bit of a framing question that I think comes up in a number of what you're going to hear from the audience, which is an old topic. And really to do with the boundaries of the spectrum. I think many of us remember when autism was considered a much rarer disease, and have heard lots of explanations of why the perceived incidence has increased, as well as perhaps the real incidence. And I wonder, as from the genetics perspective, whether you could help us largely in how we talk to our patients and their families about what it means to quote unquote have autism, and how that may have changed over the years, and how does one say that in a way that doesn't hide all the complexity, but also is comprehensible to people who are not in the field.
It is a great and challenging question. So the first thing I'll say is just to talk a little bit about the spectrum incidence in genetic architecture. So as a general proposition, the worldwide incidence of autism, as far as we can tell now from epidemiological studies, is fairly constant across the globe, actually quite the estimates. To the extent that they vary, most of the evidence suggests that those are methodological and ascertainment issues, as opposed to differences in the underlying biology. I think it's also undoubtedly the case. We know that, as you said, autism was considered a rare disorder. And in fact, I started this talk by saying, there's a very important discussion about neurodiversity. There is. And what I'm going to talk about is this subgroup of individuals, where we found large effect mutations. We're going to talk about them as pathology. That group, I think, hews fairly, more closely in general, not completely. There are individuals who carry these mutations who don't demonstrate sort of the old version of classic autism spectrum disorders, but it's definitely highly enriched, right? So for, again, with the comorbidity with intellectual disability and with epilepsy, et cetera. And there's been a move in the field to try to define what that means, because the term has gotten so broad as the spectrum has broadened, it to include, what formerly was called Asperger's or high functioning autism now is under one umbrella. And that, as we know, is an enormous range of symptoms and functioning, including blending into people who do not experience those symptoms as deficits, which is really important, right? So I think that what we trying to think about how, so I think it's important to point out that most of the expansion that we see from epidemiological studies is undoubtedly explained by what I was just talking about. The diagnostic criteria have changed dramatically. That captures a much broader group of individuals than it has previously, and consequently it's not a surprise at all that those numbers go way up. And the studies, the people have begun to try to identify this group, and they call it profound autism. That group is, a recent study showed maybe 30% of individuals with autism, meaning that 70% are on this broader spectrum. So you think, of course, that's going to lead to, if they weren't diagnosed previously, lead to this. So I would say the notion that there's an epidemic of autism, or that we need to look to environmental insults to explain that there's not a lot of evidence that, in fact, there's very little evidence to suggest that, especially postnatal exposures of any kind, are leading to increased rates of autism. Now, all that said, the epidemiological studies cannot and do not rule out that there has actually been an increase over 40 years. Whether or not that's really rooted in epidemiological factors, whether or not there are risk factors that we don't understand yet that are in fact leading to increased risk in utero, which is where we would expect probably most of the increased risk would be. We don't know that yet. So I think that's the overall picture. You don't have to explain to your patients that there's an epidemic of autism. You can confidently tell your patients that there's zero evidence for any postnatal risk factor of any kind leading to increased rates of autism, whether it's vaccines or watching television or anything else that has come up in the literature. In some ways, that's the easy part of the conversation. I think the tougher part is to tell people what it means to have autism, and that is, that is turn to you all. I mean, that's clinical art. That's, this is a huge, broad spectrum, and you're talking either with parents or with individuals about having something, again, that we define in a way that is inherently captures pathology. And now we can tell some people that they have a genetic mutation that is contributing to that. So, I think I think doesn't differ at some fundamental level from the way that we have to talk about what the difference between, sadness and major depression, or, other areas where a broad spectrum of symptoms may bring someone into our office, but there are particular challenges among particular subsets of patients that just require a different conversation. I fear that was a long-winded and very unsatisfactory answer, but that's kind of where we are. I hope that it's helpful to be very explicit, because this is going to come up repeatedly. They're going to be, you will see lawsuit after lawsuit coming in around exposure to X, Y, and Z in the first couple of years of life. Literally, when you control for genetic risk, all of those things disappear. That doesn't mean it's all genetic. It just means that there is no solid evidence for early life exposure leading to ASD.
No, I thought it's a great answer, and thank God we have the time to sit with our patients and their families and say the things you just said. That's helpful. I'll turn it back to you, Jeff, for how to pick some of the other great questions off the list.
Bettina Bernstein, hi out there. Bettina is a child and adolescent psychiatrist, and she writes in a couple questions here. First, with gratitude that you're here, Matt, and is wondering about the impact of interventions that might improve mitochondrial function, like BSM or omega-3 fatty acids, and potentially reversing genetic abnormalities, akin to thinking regarding stress reduction, helping reverse shortened telomeres, and was wondering your thoughts about that. And a follow-up question, what your impression of Bob Hendren's research regarding supplements to improve mitochondrial function, and if there's any improvement in genetic markers. Those are Bernstein.
You mean about a member of my faculty? Okay, so Bob is wonderful and an amazing clinician and important researcher and the world's most lovely man, and he recently retired. So I would say nothing but positive things to say about Bob Hendren. Okay, so what I've described for you is a slice of a slice of a slice of autism. And when you think about it, what I've told you when I described the strategy is that we had a particular way to shine a light, right? So I think there's this metaphor about a parking lot and you've lost your keys, and and so where do you look to find them? And then so we have a light post, and that light post was a variety of genetic methods that allowed us to find one subset of risk for autism. That explains in all comers maybe 10% of autism. Again, in those with intellectual disability or in girls, it may be higher than that. We don't know yet, and it's a really important question, how much that biology generalizes to what we were just talking about, to this broad spectrum of people who have autism, across the spectrum. So the reason I say that is that, well, as I said, for me personally, it's breathtaking, gratifying to be able to not only see these genetic mutations, but begin to understand what they tell us as molecular clues, that I'm also humble about the fact that at best, I'm giving you insight into 10% of what's happening here at this point, and I don't know the degree that it applies to the other 90%. So that's a long way of saying that there are a lot of very interesting and important hypotheses about biological processes that would make sense as being able to alter the kinds of processes that we see are altered by these genes. At this point, when we take a look at these genes, they don't point to, at least to the degree that we can understand them, to a variety of things that could be quite interesting. So one is, oxidative stress. When we take a look at the genes and say, if we take an unbiased look at these, do they point to a signal? Or are there a bunch of them that, that tell us that in a statistically significant way that oxidative stress is an important mechanism that's being mediated by these mutations? We don't find that. Neuroimmunology, really interesting, really important. Very strong hypotheses about the interaction of the maternal immune system, et cetera. The fact that we don't see a signal for that in the genes that we discovered doesn't mean that that is not an important biological mechanism. It just means that we don't understand it yet. So there's a long way to go to understand the biology of autism spectrum disorders. And the things that you've mentioned, mitochondrial function, we know that there are mitochondrial disorders that lead to things that overlap with autism, rare intellectual disability syndromes that also have epilepsy, also have ASD. So it certainly is highly plausible. We don't have, based on the genetics, as a strong mechanistic evidence from the genetics that we've discovered for those hypotheses. So what that means is that there's a lot of research that needs to be done apart from the work that we're doing and trying to understand what these specific genes mean, that there's a lot of work to be done to see whether or not work in those other areas, whether it's neuroimmunology, oxidative stress, cellular aging, those kinds of things, whether or not those we just don't see, but they in fact overlap with the genetic signal that we're getting. And then the last thing that I will say is that since genetics has done nothing for kids in the clinic, whether or not there's an strong ideological hypothesis that's supported by the genetics, if there are clinical trials that are done rigorously and show improvement, like that's tremendously important. And the worst thing that we could do would be to hold on seeing whether or not safe and effective interventions, or safe interventions are effective because we don't see that signal in the genetics. So that's the answer.
Thank you. Andrew, are you seeing a question?
Sorry about that. Yes, there are so many good ones. It's hard to choose.
It's nice. 7 minutes.
Let me see. I hadn't narrowed it down. How about if we go with medication selection? I don't know if you've heard this question before, Matt, but people are increasingly using some of these genetic tests for selecting medication based on metabolisms. Do you see anything on the horizon for using genetics to help with medication selection for individuals on the spectrum?
I'm going to try to be okay. So there's zero evidence that for the use of commercial genetic testing as far as I'm concerned, across all of psychiatry, including in autism. So there are panels out there, including some of the genes that we've discovered, that try to make some case that this is an important thing to know with this, with either treatment selection or expectation. None of that is true. It is the case that that drug metabolism is relevant for many of the drugs that we use. I think it's an open question so far. We've not gotten to a point where that I think clearly differentiates from clinical practice of making selection around medications, and then particularly for kids, starting low, going slow. But there are some places, Mayo in particular, et cetera, that are studying this, particularly in mood disorders. There's nothing that I've seen in autism so far that suggests that that's going to help tremendously. So as a general proposition, not terribly helpful. The sales pitches are completely unsupported, and so strong recommendation not to use them based on the notion that somehow, apart from telling you whether or not someone may be slightly likely to have more of a side effect from an SSRI, that they're not helpful in any way apart from that in dictating what kind of treatment is going to be effective in ASD.
Matt, just firing off a couple questions here. Dr. Elizabeth Ortiz-Schwartz is out there, sends in a couple questions and says, can you comment on the high parental, especially paternal age, as it relates to increased de novo mutations in individuals with ASD? And then a follow-up question, are individuals with ASD with genetic mutations are those symptoms generally more severe? You kind of hinted at that a little bit.
Yeah, although it won't surprise you, there's always a more complicated answer. Okay, so the first one was age. Okay, so we and 3 other labs discovered that these de novo mutations in the sequence of the DNA contribute to ASD and could be used to identify genes. At the same time, all of the labs identified that there was a relationship. First of all, that most of those mutations are coming from fathers. This is for sequence changes. There are other kinds of changes, copy number variation, that are more evenly distributed between moms and dads. But the point mutations, 80–90% are coming from dads, which, by the way, for the refrigerator mother hypothesis, like exactly the opposite, at least with regard to genetic risk. And then once we identified this paternal, that these were paternal, it made sense that, that's happening because sperm are turning over very frequently. And in any generation of a cell, you have some likelihood in the normal process of generating a de novo mutation. And so then it would make sense that the older the sperm were, potentially the more mutations there would be. And yes, that has been borne out. So increased paternal age increases for everyone, regardless of whether there are autism mutations or not. There's a linear relationship between paternal age and the rate of de novo mutation. And anything that increases the rate of de novo mutation is gonna increase the likelihood or the possibility that one of those mutations lands in a spot that can contribute to ASD risk. Right now, that doesn't explain a whole lot in terms of changing the effect of that is still relatively modest, even though there's a clear relationship there. So that's where we think paternal age is contributing. It's also very possible. So we think that these rare, and the majority of people with ASD who don't have these mutations have them as a consequence of an inherited variation. And so there's apart from the increased rate of de novo mutation, there are strong hypotheses about sort of the subtle social alterations leading to later, along with some of the secular changes in cultural practices around marriage, but that can also lead to this observation of increased paternal and maternal age in parents of individuals with autism. And so it was de novo mutation. What was the second one? Sorry.
Was about the difference in kind of clinical presentation of those with the genetic mutations that you've seen.
So it is the case that we looked under the lamppost and then we found these, and then we found that many of them overlap or can overlap with intellectual disability and other things that would contribute to a notion of severity. We could do an hour on what does severity mean in autism, there's a lot of variability in scales when you try to measure severity because there's not a consensus about exactly what that means. But for individuals who have, don't have language and have intellectual disability and epilepsy generally, and a lot of these genes fall into that category. It is also the case, though, that we found in all of our studies that you will find in individuals with 160 IQ and no dysmorphology one of these rare mutations. So it doesn't guarantee seeing someone who doesn't have those clinical features, but we know that those clinical features increase the yield of finding de novo mutations when we do clinical genetic testing.
Great, thank you. We have only a minute left, which is about enough time for me to say a huge thank you. So great to see you again, as always, and thanks again for joining us and teaching all of us. Andrew, thank you for being here and supporting this program and for great questions and helping me with this. And everybody out there, thanks for tuning in. We have really a great grand round series coming up in a couple of weeks with Dr. Ortiz Schwartz. So please continue to join in and tell a friend. And if you would like continuing education credits, that evaluation survey will pop up automatically in your browser, and just fill that out and send it in. So again, thank you, everybody. It's a little harsh when we just end and are all alone in our rooms, as I like to say, but it's been great being with everybody. Thank you.
Thanks, Jeff.